Oh, okay, that goes. Hey guys, so, I thought I would make some videos because of reasons that I will explain later. They all kind of fit together. And so that's what I'm doing. That's where this one starts, and I guess this is what we're gonna do here. So, I am an intern at the Uintah Basin, which is where I live, USU campus. I am interning in the biology and neuroscience lab. That's what I do, and I started in June, and a couple weeks ago, and I will be done on August 1st. And so that's what I do. I'm still just at the beginning stages of it, but one of the things you have to do when you work in a lab is leave a journal of everything you do, and so that you have a resource that you can look back on, and also for other people to know what's in what you did and your experiments and your thoughts and all that stuff. And while I am doing that, and it's great, I also figured, hey, maybe I could make a video blog about it or a blog, or just videos, I guess, about it, and then that way I could maybe remember more, or if anybody has any interest in it, just watch my videos. Here you go. So, I guess I will explain what I'm doing with this internship. I am studying blue-ringed octopuses, which is super awesome. They are the guys that are really toxic, and they change their shape, or their color. Invertebrates change their shape, but, and the thing that we're most interested in is actually the toxin that they have, because it is the same toxin that is found in newts in Oregon, and pufferfish, and frogs in Central America. So it's a pretty big deal, and it's called tetrodotoxin, or short for TTX, because sometimes I can't say tetrodotoxin, so I just say TTX. And what it is, just as this basic overview, is a neurotoxin, and it affects the way your neurons work, because what they do is they let in electrons, sodium ions, in and out, and that's how they move electricity among their cells to send messages and release neurotransmitters, all that kind of jazz. And it's through these sodium channels and sodium ions, and they move through sodium channels, which are these proteins in cells that control the access in and out of sodium. So what the tetrodotoxin does is it actually gets in the way. It's kind of like it basically dams up the channel so that nothing can come in and nothing can come out, and then a little while later you die because you're completely paralyzed, and it's terrible. It's super, super toxic. I think like one average-sized newt, it's probably that, an average-sized newt, has enough TTX to kill like 5 full-grown men. It's like a big deal. You don't mess with that crap. And so what we're studying is actually, back to those newts, they actually have this coevolutionary relationship with garter snakes in the area, where the newts get more poisonous and the snakes get more resistant. So it's this kind of constant poison resistant, poison resistant, poison resistant. So what we're trying to find out is how that can work, because TTX is in animals, which are kind of weird, where they have it in their entire body, like it's just everywhere. So you can't get any part of them, which is kind of the opposite of many venomous things. Because like for snakes, for example, they have their poison contained in their special glands and their fangs, and it's not throughout all of them. So that's where the thing is, because this horribly toxic thing that destroys all neural stuff is in fact found in all of the body parts of these animals, and we're trying to find out how their sodium channels are different than everybody else's, because, and you'd think that would sound really easy, right? Like, we'll just run a couple experiments, but there are actually like 8 or 9 different sodium channels, like 1.1, 1.4, 1.8, and like 1.4 is cardiac muscle, is your heart tissue, and it's resistant to TTX, and I think 1.6 is sort of pain receptors, and it's sensitive to TTX. So within the channel there are different versions of the channel that react differently. And actually in invertebrates and the blue-ring octopuses that I'm going to do, they have 2 different sodium channels. They have like 1.4 and then they have like 2.8. Like they have 1.1 and 1 and 2s. And so the thing is, the second sodium channel has not been studied very much. So that's what we're going to do. We're going to study that. And how, you may ask? Well, in my experiment, in my lab that I'm a part of for the summer, which is fantastic. If you ever have the opportunity to do that, you should. We've got some blue ringed octopuses. They're coming. I think they're being shipped in from Indonesia. I think that's where they live. Well, I know that's where they live, but I think that's where they're being shipped in. And they unfortunately do not live for very long in captivity. Partly because they're only like 8 inches long. But they're not very big. And so they're going to be shipped in, and we're going to study them for a little while they're alive, just to get a basic understanding, because they're invertebrates. Invertebrates are weird, especially because what we're doing is dissection later. Sorry. But after they die, we are going to process them, which is scientific lingo to make ourselves feel better about dissection. And then we're going to study their various tissues, because basically what an octopus is a bundle of nerves. That's basically what it is. And so for blue rings to have tetrodotoxin, they must be super evolved in how they live. So what we're doing is figuring that out, and then we're going to run a bunch of tests on different tissues that they have, and then afterwards we are going to get their RNA out, which is super hard because RNA is more unstable than DNA. I don't know if that's something you know, but nowadays, remember that RNA is the first step, and it's more unstable than DNA. It's a lot harder to get and study, and so we're going to extract that from their cells, and then we're going to actually make cDNA, copy DNA from that, so that we can study their entire transcriptome. We can actually do that at my campus that I'm at now, because it's like millions of dollars and super hard. So we're actually sending it out to have somebody else do it for us, and then they are going to send us back, like, I think my professor said, like 2 DVDs worth of data. And we're going to get it back, we're going to start studying that, and we're going to look at like similarities and differences, and we're going to have a transcriptome of their entire genetic thing, genome, I guess, because oftentimes that's the best way to study the actual stuff that organisms do is their transcriptome, because sometimes weird stuff happens, and so it's best to know how they're supposed to work based on their genes. So that's basically what I'm doing, and I will be posting videos every so often. I haven't decided, maybe it'll be like every 2 days or something, but just to keep everybody updated, and they will be probably the same length, just because that's what I feel like I should do. So I will see you guys later in a couple days, and we will have some data. Hopefully we'll have our octopuses. We don't have them yet because they're being shipped, but hopefully we'll have them, and I will be able to tell you guys something awesome for all of you viewers. Who knows how many you are, but if you have any questions, always just ask, put them in the comments or anything, and I will try to get back to you and answer them. I can't make any promises, but I will definitely do my best. So it was great talking to this camera and pretending that it was real people listening to me, and we'll see you guys later.